The overall goal of the following experiment is to detect modulation of G protein-coupled receptors by potassium channels, utilizing label-free resonance wavelength grating optical biosensors. To achieve this, untransfected HEK293 cells and HEK293 cells stably expressing rat sodium-activated potassium channel SLAC-B are seated onto a 384-well optical biosensor plate and allowed to incubate overnight. Next, a baseline measurement of mass near the plasma membrane is performed, and agonists of endogenous G protein-coupled receptors, or GPCRs, are added. As a final step, the change in signal resulting from GPCR activation is measured, and the potassium channel modulation of this response is calculated. Ultimately, this method can help answer key questions in the fields of neuroscience and cellular signaling by quantitatively measuring signaling mechanisms between G protein-coupled receptors and potassium channels.
The main advantages of using this technique over the methods that currently exist to look at the interactions between channels and proteins, for example, co-immunoprecipitation assays, are that the assay can be carried out using living cells. It doesn't require the use of exogenous labels, which could alter the structure of the interacting protein. And finally, within the living cells, you can actually follow the interactions in real time.
Though this method can provide insight into signaling between GPCRs and potassium channels, it can also be used to study cellular responses resulting from ligand-induced changes in cell adhesion and spreading, toxicity, proliferation, migration, and any changes in the proximity of the plasma membrane to the optical biosensor.